Hey guys, today's lesson is about protein synthesis, which is how proteins are made. We've talked before about the fact that proteins are made from amino acids and that this happens in the ribosomes, but we're going to go in through the full process today. Okay, so in this lesson about protein synthesis, we're going to be going over the relationship between DNA, genes, codons, amino acids, and proteins. We're going to briefly talk about the characteristics of the genetic code and what the central dogma of protein synthesis is. Then we're going to go over the two different parts of protein synthesis, which is the making of mRNA during transcription, and then the translation process, where we combine amino acids to make the polypeptide. So you should just know this whole process altogether. So we've talked before, like I said, about the fact that proteins are made up of amino acids. We learned in Bio 20 that a chain of amino acids is called a polypeptide, and then when you fold that polypeptide, you get the protein. Gene expression refers to the conversion of a gene into a specific trait by producing a specific protein or polypeptide. So the genetic code is really just a sequence of nucleotides on each strand of DNA, and then the genes are different segments of that DNA that carry coded information for one protein. And then we talked about before when we were going over mutations, but codons are 3 consecutive nucleotides of mRNA or DNA that code for a specific amino acid. Each codon will code for an amino acid, and then all of those amino acids will be linked together to make the protein. There are 20 different amino acids that make up proteins. The genetic code is read one codon at a time that says which amino acid goes next in the polypeptide lineup, and then the combination of codons that make up a gene will code for the combination of amino acids that make a protein. So when we say that a gene codes for a trait, we're saying that it codes for the protein associated with that trait. So for example, the gene for eye color is really coding for the protein that makes eyes a certain color. So while some of the genetic code will code for a specific protein, there are also non-coding regions of DNA, which is just kind of junk or gibberish DNA that doesn't code for anything. And actually about 90% of the human genome is this junk DNA. Of the remaining 10%, some of it is structural, which codes for the production of proteins, and then about 3% is regulatory gene, which regulates structural genes by turning them on and off. There are 3 characteristics of the genetic code. The first is that it's redundant, which means that one codon, multiple codons can code for the same amino acid. So there's only 20 amino acids, but there's way more codons than that. So you'll have one amino acid that might have like 4 or 6 different codons. This is really important because if there is a point mutation, it means that it might not actually change the amino acid that it's coding for, even though the codon changed. That's what we call a harmless or an invisible mutation. The DNA has changed, so it is mutated. However, it doesn't have any effect because the protein's still the same. The amino acids that make the protein are still the same. The genetic code is also continuous, meaning that you read it the same way you would read a sentence, just like one codon after another, just like you would read a sentence one word after another. And lastly, that the genetic code is universal. And the reason that it's called universal is because almost all living things use the exact same genetic code, the exact same nucleotides to code for and build proteins. This is the central dogma of protein synthesis. It's essentially the idea that you convert DNA into mRNA and then mRNA into amino acids, and that's how you make a protein. So the first part of that, converting DNA into mRNA, is called transcription. And then converting the mRNA into amino acids is called translation. And like we said before, this is done at the ribosomes, which are in the cytoplasm of the cell. So we'll start with transcription. Again, that is when you take a gene from the DNA and turn it into a strand of mRNA. The mRNA strand will be complementary to the DNA strand. The DNA is in the nucleus, it can't leave the nucleus, so transcription must take place in the nucleus. It is initiated when RNA polymerase binds to what's called the promoter sequence at the start of a gene within the DNA. The promoter sequence is just a sequence of nucleotides that are right in front of the gene. It's sometimes called the Tata box because it usually starts with a bunch of T's and A's in a row. And then within that promoter sequence, there's something called the initiator codon, which is TAC. So the RNA polymerase will bind to the Tata box, and then it will start actually building the new mRNA at the initiator codon, the TAC. So yeah, the DNA has to be uncoiled, the two strands are separated, just like when we're replicating DNA, only this time, instead of DNA polymerase 3 building a new strand of DNA, RNA polymerase will build a new strand of mRNA. Now, because RNA is only single-stranded, you're not going to have a leading strand and a lagging strand because you're only going to be making one strand of the mRNA. And so the strand of the mRNA is built off of the antisense strand of DNA, which is the one that goes 3' to 5'. And the reason for that is so that the mRNA can be built 5' to 3'. Just like when building DNA, we can only build new molecules of DNA or mRNA in the 5' to 3' direction. The sense strand of DNA will be the opposite one to the antisense strand, so it'll be the 5' to 3'. So the RNA polymerase will just keep building the mRNA by adding one new nucleotide at a time until it reaches what's called the terminator sequence or termination sequence, also sometimes called the termination codon. It is the codon that tells the RNA polymerase that it has reached the end of the gene and can stop building the strand of mRNA. After that, the mRNA will detach from the DNA. The DNA will close back up together, reforming the hydrogen bonds between the nitrogenous bases, and then the mRNA will be processed before moving to the cytoplasm. The processing part of this is essentially just getting the mRNA ready to leave the nucleus. Two things need to happen in order to protect the mRNA on its trip out of the nucleus. One is that what's called a poly-G cap is added to the 5' end of the mRNA, which is just a whole bunch of G's in a row. And then we also add a poly-A tail to the 3' end, which is just a bunch of A's in a row. And those protect the mRNA from enzymes in the cytoplasm that could destroy it. And then we also cut out things called introns from the gene. So like we said, there's a lot of junk DNA in our DNA. And so part of the gene will be that like junk DNA. And that junk part is called the introns. We cut them out, and then the remaining parts that are the useful information or the coding areas have to get reattached to each other. So the exons are the parts that get left over, they get reattached, that's the important part. And the introns are the stuff that we take out, which I know is like backwards from how it should be, but the introns get taken out, the exons stay in. Okay, so this is kind of what the process looks like. Again, according to the central dogma, transcription first, which is going from DNA to our mRNA, which is this pink strand, and then the mRNA to the amino acids that make the polypeptide is the translation portion. So that's what we're going to move on to next. So again, the mRNA is processed. It is going to leave the nucleus and go out into the cytoplasm where it can meet up with a ribosome. There are two different subunits of the ribosome. There is the small ribosomal subunit that actually reads the mRNA, and then the large ribosomal subunit joins the amino acids together to make the polypeptide. So the ribosome is going to recognize the mRNA and grab onto its poly-G cap. And then starting with AUG, which is the start codon on the mRNA, the ribosome is going to start reading the mRNA one codon at a time. The first codon of the mRNA is going to go into what's called the P site of the ribosome, like the letter P. And then the ribosome will, from that point, start moving down the mRNA in the 5' to 3' direction, again, reading one codon at a time. Every time a codon is read, a tRNA molecule is going to come and bind to the codon of the mRNA. The tRNA is going to have kind of like an anticodon, which is the complementary pairs to the codon. The tRNA comes over, it is going to be holding up the amino acid. And then when the next tRNA comes over holding up its amino acid, the two amino acids will form a bond between them. So like I said, the first codon is going to be in the P site, so that's where the first tRNA goes. The second one is going to go into the A site, and then they're both going to like shift over like it's a conveyor belt. The one that's in the P site is going to leave. The one that was in the A site goes to the P site, and they all just kind of bump down. And that'll just keep continuing with peptide bonds forming between the amino acids until we get to the stop codon. The stop codon is just the very last codon in the gene. And again, it tells the ribosome that it's done. So this is kind of what it looks like. This big purple blob thing is the ribosome. This boomerang shaped thing is the tRNA. It's holding up the amino acid. And then this green part down here is the mRNA. So right here, we have AUG, that is the start codon on the mRNA. The UAC is the anticodon of the tRNA because A and U are complementary and G and C are complementary. So the tRNA that has the right anticodon comes in and holds up its amino acid. Then the next codon of the mRNA is read, so in this case, GCA. The tRNA with the right anticodon, so in this case, CGU, will come over. It'll go into the A site, and it'll hold up its amino acid. And then a peptide bond will form between those two amino acids. The tRNA from site P will leave. The one from site A will move over to site P, and then a new one will come into site A, and that'll just keep on continuing. By the way, this shape up in the uppermost corner is a more accurate representation of what a tRNA molecule looks like. It doesn't really look like a boomerang, it's more of like a clover. Okay, and then once the ribosome reads the stop codon, that says we've reached the end of the gene, we can stop building this protein. The ribosome subunits will break apart and release the mRNA and the polypeptide chain, and then the polypeptide chain will get folded up into a protein. If you remember back from Bio 20, the order of the amino acids in the polypeptide chain is the primary structure of the protein. Then when you fold that polypeptide and create different hydrogen bonds in the shape of alpha helixes and beta pleated sheets, those are the secondary structures. Then the alpha helixes and beta pleated sheets can bond to each other to make the tertiary structure. And then if you have a whole other protein come bond with your protein, that is a quaternary structure. Proteomics is the study of proteins produced by a given genome. There is a lot of research still being done on how proteins fold and trying to determine the folded shape of proteins and stuff like that. Because once you know the shape of a protein, you can start designing drugs to interfere with those proteins, which is really useful when you're treating different diseases and disorders and stuff like that. So this is what I was just talking about with the 4 different structures of a protein. So the order of amino acids is the primary structure. Get some hydrogen bonding to create alpha helixes or beta pleated sheets. Bond those to each other to form the tertiary structure, and then the quaternary structures if you have more than one protein attached to each other. With this, anytime you're going from a DNA triplet to an mRNA codon to the amino acid, what you're really doing is saying, okay, if the mRNA codon is TAC, then the mRNA must just be the complementary bases to that. So complementary to T is A is normally T, but with RNA we don't have T, so A is U, and then C is G. So that is the mRNA. And then in order to find the amino acid, you actually need a sheet. It's like a chart, which you will be given. And if I forget to post it, let me know, but I'll show you what that looks like. Okay, so this is the chart that you need in order to go from an mRNA codon to an amino acid. So for instance, if I just read that that amino acid is AUG, I go on here and AUG is methionine, or MET for short. Okay. So yeah, I'll post this and then you guys can practice filling this out. Yeah. So you should have, for the second one, let's see, CCA would be GGU. We just did that one. GGG would be CCC. TAT would be AUA. Again, I'm just doing the complementary base pairs. So A is complementary to T, C is complementary to G, and then ATT would be UAA. And then we already said that that is, oopsies, methionine. GGU is glycine. CCC, need to move this. CCC is right here, it's proline. AUA is right here, isoleucine. I'm just writing the short forms to save time. TGG, oh, there's no such thing as T's in mRNA. UGG. UGG is tryptophan, which is the thing in turkeys that make you sleepy when you eat them. And then UAA, which is the one at the bottom here. UAA is actually special. Actually right here, UAA is a stop codon or a terminator codon. So it doesn't actually code for an amino acid, it just says that we've reached the end of our mRNA. Okay, so that's the answers that you should get for that. I know that this was a lot to wrap your heads around, and that especially the translation portion is complicated. So I am gonna like do a little drawing thing for you right now to go over this again. But of course, let me know if you have any other questions after I do that. And what else? I think that's it. Okay. Okay, so this is the transcription portion of protein synthesis. Again, the DNA molecule is going to be opened up. The two different strands are going to be separated. The strand that goes in the 5' to 3' direction, which is this top strand here, is called the sense strand. And then the strand that goes in the 3' to 5' direction is the antisense strand. The antisense strand is the one that the mRNA is going to use as a template. So again, this is going to all start at a little something called the TATA box, which is right there. That is the promoter sequence. That is where the RNA polymerase would bind onto in order to start building this mRNA. And then the initiator codon is right here, the TAC. So that's where it actually starts building the mRNA. And then it'll start by placing one complementary nucleotide at a time the whole way along. So A is complementary to T, U to A, G to C, C to G, etc. And it'll keep doing that until it reaches the terminator codon, which is right there. Okay, so we've got our initiator codon where the mRNA starts, the terminator codon where it stops being built, and the promoter sequence, which is where the RNA polymerase would bind. The RNA polymerase is the molecule that actually builds the mRNA. Again, mRNA, just like DNA, can only be built in the 5' to 3' direction. Then we process the mRNA by adding our poly G cap and our poly A tail to protect it on its trip out of the nucleus into the cytoplasm. And then we get into transcription, which happens at the ribosome, which is this lovely guy. The mRNA will come in, and the ribosome is going to start reading the mRNA, like so. The first codon, and each one of these things that I've circled is a codon. The first codon is our start codon, and it's going to go into the P site of the ribosome. And then the ribosome is going to read the codon AUG. And that is going to cause our tRNA, which is this guy, to come over. I know that it's this tRNA because it has the right anticodon. So I've circled in pink the codons. This part down here, the UAC, is the anticodon on the tRNA, which is this clover-shaped thing. So that's going to come, and the tRNA will enter the P site, and the anticodon will bind to the codon. Then the next codon is going to be read by the A site of the ribosome. So CGU, that's going to cause this tRNA molecule to come over because it has the complementary anticodon. C goes with G, G goes with C, U goes with A. So this tRNA is going to go into the next site, like so. Then what's going to happen is these two little boxes here are the amino acids. These amino acids are going to form, oopsies, a peptide bond that will hold them together. Once that peptide bond is formed, the tRNA is going to let go of its amino acid. I didn't mean to flip upside down. And now this tRNA that's no longer holding its amino acid will go to the E site, which is the exit site, and leave. And it can go leave and pick up another methionine, another met. Then what's going to happen is the mRNA, still bonded to this tRNA molecule, is going to shift down. So that's going to shift over to the P site, like so. Stop sticking to me. Okay, so now that tRNA is in the P site, the A site is open, so the A codon is going to be read, so AGG, which is going to call over this tRNA molecule because it has the right anticodon. The codon AGG is complementary to UCC. So this guy is going to go into the A site. And once in there, we're going to have a peptide bond form between those two amino acids. Okay, once this new peptide bond forms, this tRNA can let go. It'll go to the E site and exit. And then this guy, still attached to the mRNA, will move over to the P site, which then opens up the A site. So that'll be read CCU, which will call over this tRNA because GGU is CCU is complementary to GGA. So this guy is going to enter the A site. Why is this one so much taller than all the other ones? A bond will form between those amino acids. Once that bond forms, this amino acid can let go of the tRNA. The tRNA will enter the E site and leave. Then the tRNA, still bonded to the mRNA, will all shift over to the P site, like so, which will cause this codon to be read by the A site, UAA. Now UAA is our stop codon, which means it doesn't have a matching tRNA because there's no amino acid that goes with that. So as soon as we hit the stop codon, we are at the end of our polypeptide. So this tRNA can leave, go to the exit site and leave, and then our ribosome is going to release the mRNA, and it's going to release the polypeptide. Okay, so this is our polypeptide. This would fold up to become our protein. The mRNA will just get broken down. It's done its job. The ribosome can get reused next time a protein needs to be made. each of these tRNA molecules would go back and pick up another one of their matching amino acids. Again, remember, each one of these tRNAs has a specific amino acid that it can carry based on what its anticodon is, or more specifically, what the codon of the mRNA is that it matches. That's what we use our mRNA to amino acid chart for. But yeah, that's it. That's how we form the protein. So transcription first, which is that process, and then translation second. They come in alphabetical order. Okay, let me know if you have any questions.